In lecture 6, we're going to be looking at the anatomy, the structure of the spinal cord as well as the brain. We're going to learn about different parts of the brain as well as the function associated with each of these parts. So by now, we should know that the central nervous system, or the CNS, consists of the brain and the spinal cord. What makes up the brain and the spinal cords are actually two types of matters that we call gray matter and white matter. So the white matter is literally white in color, more like an ivory color, I would say, because it contains myelinated axons. Okay? So we learned in lecture 5 that some axons are going to have myelin on them to help them increase the conduction speed. In fact, a majority of the axons are going to have myelin on them, and they make up the white matter because the myelin actually gives it a shiny white appearance. Okay, so within the central nervous system, a bundle of axons is what we call tracts. Okay, so these tracts are important for conveying information, carrying signals from one place to another, and that's essentially what white matter is. On the other hand, we have gray matter, which basically is everything else. So there are the cell bodies, there are some glial cells, there are some short non-myelinated fibers. So not every axon is going to need to have myelin on it. If it's really, really short, then having that myelin is not really going to increase the speed significantly. So some of these axons are what we call naked axons. They're exposed. They don't have any myelin on them, and that would be part of your gray matter. Now, we want to protect the central nervous system as much as possible because the brain is very precious, obviously, and we want to avoid any kind of damage. So besides the skull, which protects the brain, and the backbone, the vertebral column, which protects the spinal cord, so in addition to those bony structures, you also have something called the meninges. Okay, so the meninges is basically a three-layer membrane that protects the brain and the spinal cord, and it's going to be in direct contact with these structures. You might not have heard of meninges before, but you probably have heard of meningitis, right? So meningitis is a disease where it's a condition where the meninges is inflamed. We're going to talk more about that later. But for now, the meninges, there are three layers to it. The outermost layer of the meninges is something called the dura mater. Okay, so this is not a spelling mistake, it's just a single T, dura mater. And this dura mater is, as the name implies, very, very durable. It's tough, it's white in color, and it's a kind of fibrous connective tissue that lies right next to the skull and your vertebrae. Okay, so the skull is going to be in direct contact with the outermost layer of the meninges, which is the dura mater. Okay, and again, vertebrae is basically your backbone. So let me put it here for you, backbone. The dura mater itself is actually made up of two separate membranes layer. Okay, and these two membrane layers are typically stuck together, and they only separate in several locations within the brain. So in areas where they do separate, they form a cavity, an empty space that is called the dural venous sinus. I'm going to show you a picture of that in a few slides later. But for now, this is the space where you will be draining excess cerebrospinal fluid. Okay, so in the brain and the spinal cord, it is surrounded by this fluid called the cerebrospinal fluid that will help protect the brain from having concussions, for example. And we actually will go into more details about the cerebrospinal fluid later on. But we need to have a place to drain these fluids. Otherwise, they just kind of built up in the head, and they will drain in the dural venous sinus. There are a lot of interrelated concepts here, so it's hard to talk about one without talking about the other. So, I'm just try to give you a brief overview of some of these things first, and then we will add the details to it later on. So again, right, the dura mater, the outermost layer, which typically has two layers that are stuck together, but at some places they separate and form these dural venous sinuses to drain the cerebrospinal fluid. So beneath the dura mater is another layer called the arachnoid mater. Okay, arachnoid means spider, right? Okay, it's actually written right here. So these are spider web looking structures. Is going to give a little bit of shock absorbing capability if you bump your head, for example. And you know, there are these empty space in between the spiderweb structures, and that is going to be filled with the CSF. As a loose analogy, you can think of the your mattress. Okay, so like, you know, you got the side that you are sleeping on, right? So that would be like the dura mater, and then you have the spring coils in the middle, and that would be the arachnoid mater. And then there is the underside, right? So the underside is going to be the pia mater. Okay, and of course between the spring coils there are a lot of empty spaces. So between the spiderweb looking structure in the arachnoid mater, that empty space is going to be filled with the cerebrospinal fluid. Okay, so the inner layer, the innermost layer, the deepest layer, is called the pia mater, and this is the layer that is going to be in direct contact with the brain or the spinal cord, depending which part of the CNS you're looking at, and they are closely following the contours of the brain and the spinal cord. The contour just means, you know, if there's a bump or groove, then the pia mater is going to be following those shapes as well. The best thing that I could think of to help you understand this is if you buy like frozen meat or something at the supermarket, sometimes they are vacuum sealed, right? And so the shrink wrap that goes on top of the meat would follow the contours of the meat, right? So that shrink wrap is kind of like the pia mater. It's completely stuck to the brain and the spinal cord, and then it follows all the grooves and the bumps in the brain and the spinal cord. So in this diagram, you can see this is the coronal section. It's separating the body into front and back, and we are focusing at this here at the top of the head. We're zooming in to see the various structures. It's probably better that we label this together in the workbook. So please go and find this diagram in lecture 6 of the workbook. So here we go. First of all, we have the skin. Okay, so these are hair, right? So at the bottom of it, that would be the skin of the scalp. I'm just gonna put skin here, and underneath skin, we have what's called hypodermis. Okay, hypodermis. Right, so these are not particularly important. It's just like typical stuff. Now, underneath the skin and the hypodermis is where you have the skull. Okay, so this here, that's the bone that protects your brain. Okay, so that is the skull. Okay, it's actually pretty hard. The skull. I remember reading an article about a pair of scissors falling off out from the sky, and it actually impaled into a pedestrian's head, but the skull was actually able to stop the scissors from penetrating into the brain. So there was this bizarre picture of the lady with the scissors stuck in her head and being rushed to the hospital. So very, very strong stuff. Now beneath the skull, there is going to be the dura mater. Okay, so you can see that's the dura mater, and that's also the dura mater, right? Remember, the dura mater is two layers fused together, but at some locations in the brain they will separate. So here, kind of at the top of the brain, that's one part where they are going to separate, and when they separate, they are going to have a cavity, okay, a space, empty space. Okay, so that's empty space is shown here, and we're gonna label that in a second. But first, let me put it here. Okay, this two layers, that is the dura mater. Dura mater. Okay, very durable stuff. and it's going to be in direct contact with the skull in the case of the brain, and it's going to be in direct contact with your backbone, with your vertebrae in the spinal cord. Okay, and again, they are two layers fused together, but sometimes they would open up, and when they open up, this empty space is called the dural venous sinus. Okay, so a venous sinus in the body is basically a location where you drain fluid. You have venous sinuses in other places of the body. This one is found in the dura mater, so that's what we call the dural venous sinus. And I'm just gonna put it here to remind you the dural venous sinus is where you will drain excess cerebrospinal fluid. Okay, CSF. We haven't really talked about CSF yet, but we will. But suffice to say, you produce them continuously throughout the day, so you must have a way to drain them. And this is one location to drain them, at the dural venous sinus. Okay. I'm gonna go ahead and do the innermost layer first. Okay. That is the pia mater. Okay. And the pia mater is basically what I'm drawing here. Okay. It's going to be following the contours of the brain. Okay, so it's going to follow all these grooves and is going to be stuck to the spinal cord as well. Okay, of course you don't see the spinal cord in this diagram, but you can imagine it doing similar things as what I'm drawing here. Okay, so that is your pia mater. Okay, pia mater. Okay. Between the dura mater and the pia mater is going to be the arachnoid layer. Okay, so the arachnoid layer are these spiderweb looking stuff, right? Arachnoid mater. Okay, and it's also going to be filled with CSF. Okay, so this arrow is actually pointing at the space. The spaces between the spiderweb looking structure, right? Remember I said those springs in your mattress, there are a lot of empty spaces there. So that empty space is called the subarachnoid space, right? Subarachnoid space. So that label is subarachnoid space, right? And is filled with CSF. Okay, and then all the extra CSF will be drained in the dural venous sinus. So I'm just gonna, put some blue here to represent the CSF. Okay, and we're gonna drain them here as well. Okay, there we go. All right, so together the dura mater, the arachnoid mater, and the pia mater collectively, these three things makes up the meninges. Okay. Here, this is the actual brain, and you can see this is the darker color, and that is the gray matter. Gray matter. And then inside it's lighter, so we call that the white matter. Okay. So for the brain, the gray matter is on the outside, and the white matter is on the inside. For the spinal cord, it is actually the other way around. Okay, so for spinal cord, it is white on the outside and gray on the inside. We probably should write that down for now, to so that you kind of remember it. So for brain, it is typically gray out and white in. Okay, there are some exceptions, and we're gonna get to that later. Okay, but for the most part, it's gray matter outside, white on the inside. And for the spinal cord, it is white on the outside, white out, and gray on the inside. Okay, so just kind of remember that for now, and we're going to come back to it later on. So that's the diagram. So the CSF or the cerebrospinal fluid is another protection for the CNS, and the CSF is formed by a type of glial cells called the ependymal cells. Okay, so the glial cells, we learned that in the last lecture. They are supporting cells of the nervous system. We learned, for example, the Schwann cells will form the myelin. So here is another type of glial cells, the ependymal cells, and they will form the CSF. Now, these ependymal cells are found in structures called choroid plexus. All right, so here is a choroid plexus. Here's another choroid plexus, and this is a frontal view of the brain. If you cut the brain into front and back, you will see two big empty chambers here, and they are two of the four ventricles that you have in the brain. Okay, so the choroid plexus are found in all four of the ventricles, and they're kind of stuck to the wall. And within the choroid plexus is where you will find the ependymal cells that is responsible for producing the CSF. So the CSF that are produced by the choroid plexus will fill up all the brain ventricles, and again you have four of them. I'll show you a picture of that coming up. And in the middle of the spinal cord, you actually have another empty channel that goes from the neck all the way to the lower back, and that is called the central canal, right? And that is also filled with CSF. Earlier, we talked about how CSF will fill up the subarachnoid space. That's basically the space in between the arachnoid matter and the pia mater. And also, excess CSF will be drained in the durovenous sinus, and it's just going to go back into your cardiovascular system afterwards. So to keep it simple, CSF is basically a clear tissue fluid, and it's derived from blood. So it's very similar in composition to the blood plasma. There is a bit of control as to what goes into the CSF and what doesn't. We're going to talk about that coming up, and it forms a protective cushion around and within the CNS. Basically, it creates a buoyancy force that will allow the brain to kind of float in the skull, right? If you move your head forward suddenly, because of inertia, your brain is gonna float backwards. Okay, and during very forceful collisions, such as like a contact sports, or if you fall and hit your head, it's possible that your brain is going to hit the inside of the skull. And by having a layer of CSF around it, it basically prevents a concussion from happening. Okay, it kind of cushions the brain to prevent it from hitting the inside of the skull. So the composition of CSF, as we just mentioned, is very similar to that of blood plasma, because after all, that's where they came from. But we do want to regulate the contents within the CSF, because the CSF is going to be in direct contact with our CNS, central nervous system. We don't want some harmful substances to gain access to our brain or spinal cord, and so the content of the CSF, the composition of the CSF, is tightly regulated by something called a blood-brain barrier. Right? We talked about the BBB, blood-brain barrier, earlier when we were discussing dopamine and Parkinson, right, in the previous lecture. But here's more detail. The BBB is formed by yet another type of glial cells, which is called the astrocytes. Okay, and the BBB allows basic stuff like water and some gas and some lipid-soluble molecule to go through by passive diffusion, and then things like glucose and amino acids that we need for crucial neural functions will be going through by selective transport, and then it will screen out a bunch of stuff that might be toxic to the nervous tissue. So here is an astrocyte, and you can see the astrocyte actually have a lot of these projections coming out from it, and they are kind of like the tentacles of an octopus, and there is a little suction cup at the end that would go around the neurons on one side, and then it would go around the blood vessels on the other side. Okay, so you can see the capillaries is entirely covered by these suctions, and that's basically what forms the BBB. Okay, the BBB is not like a membrane that you can cut out and kind of see physically. It's more like a microscopic barrier formed by these astrocytes. At this point, I think maybe it's good to just quickly review all the all the glial cells that we've learned so far. So we learned about the Schwann cells, right? Schwann cells and the oligodendrocytes, and they are the one that forms myelin. Okay, myelin. All right. Two or three slides ago, we learned about the ependymal cells. Ependymal cells, and they are the one that will form. They forms the CSF, and now we have the astrocytes, and they form the BBB, blood-brain barrier. Here is a picture that shows you, it's like a see-through brain that allows you to see the ventricles of the brain. So you can see this is the lateral ventricles. You have two of them, okay, one in each of the hemisphere. A hemisphere is basically half of a brain. So this is looking at it from the front. It forms like a horn-shaped structure. So if we go back to here in the frontal section, it looks like they are just a triangular chamber. But if you look at it from the side, like in this picture, you can see actually is quite big. Right. So let me just skip to this 3D models to show you a little bit. I post a link to this 3D models on eCentennial, so you can check that out later on. So this is half of a brain, and you know whenever you look at the mid-sagittal section of a brain, you will always see this band. And so this band is called the corpus callosum. It's white matter, and you know it basically allows the two halves of the brain to talk to each other. But what we want to focus on right now is underneath this corpus callosum is going to be your lateral ventricle. Okay, so we're looking at the right half of the brain. You know, this is the right half of the brain. I'm looking at it from the back now. Now we're looking at it from the medial side, and so this is going to be your right ventricle. Okay, you can only see a little bit of it because much of it is actually hidden within the hemisphere. So if I were to isolate this, right, then you can see this is the lateral ventricle. It forms like a C shape, kind of with a tail there at the end there. Okay, so that is filled with the CSF, cerebrospinal fluid, and then it's going to drain into the third ventricle. So here is the third ventricle, and then from the third ventricle, it is going to drain into the fourth ventricle. Okay, so there's a little duct that connects the third and the fourth ventricle, and the duct is called the cerebral aqueduct. Okay, so let's go back to the diagram here and talk more about it. Right, so you can see the lateral ventricle, and on the walls of the ventricle is going to be these spongy structure, the choroid plexus, right? I'm going to label it for you. Choroid plexus, and that's where you will find the ependymal cells, which will be making the CSF. So once you make the CSF, it's going to fill up the entire horn, and then it's going to drain into the third ventricle. Okay, so the third and the fourth ventricles are kind of found along the midline of the brain, and to drain into the third ventricle, you will have to go through a tiny hole here. Okay, so there is a tiny hole here and a tiny hole here. A hole in the body is called a foramen. Okay, foramen. Right, it's kind of like FO and then ramen, foramen, and this hole, this foramen, is found between the lateral ventricle and the third ventricle. So it's the interventricular foramen. Interventricular foramen, a hole that connects two ventricles together. Okay, and from there, it's going to go through the cerebral aqueduct, right? The cerebral aqueduct into the fourth ventricle. Okay, just let me show you one more time quickly in the 3D model here. Right, that's the third, that's the aqueduct, and that's the fourth ventricle. You can't really see the foramen here because you cut the brain in half, but you know it will be where the CSF is going to be coming out from the lateral ventricle into the third through the foramen, which is somewhere here. Okay, you're not going to see this big huge gap, right, when the two half of the brain are actually fused together. You only see it again because we cut it open, right? Okay, so here is another view. Here's another view This is an actual brain, and this time we're looking at it from the we're looking at the left half of the brain. So again, you see the corpus callosum. You always see the corpus callosum in a mid-sagittal section of the brain. That would be the lateral ventricle, the left lateral ventricle. That's what LV stands for. There is the third ventricle, and then here is the fourth. Okay, fourth ventricle always looks like a little triangle thing. Okay, and connecting the third and the fourth is going to be this duct. Okay, the cerebral aqueduct. SA is just another name for it. We're not gonna talk about it. Okay, and here is the foramen, the interventricular foramen, where the CSF will drain out from the LV into the third. And again, FM here is just another name for the foramen. Okay, so we're gonna stick with the interventricular foramen. So from the fourth, it's actually going to continue to drain down to the central canal of your spinal cord. Right, so you cannot see it in this picture, but the spinal cord would be right there, and in the center of it is going to be an empty space called the central canal. And that's where the CSF will continue to drain down. Okay, all the way to the bottom of your spinal cord. So let's try to summarize this thing we have been talking about. See if it makes any sense. So basically, you would have the blood, okay, the blood, and from the blood we are going to be creating the CSF from it. Okay, so the cord plexus, the ependymal cells in the cord plexus will filter the blood, and it's going to be creating CSF. Okay, so we are going to have cord plexus in several places. Okay, we're gonna have it in the lateral ventricle. Okay, and you have two of them times two, one in each of the hemisphere. You're gonna have cord plexus in the third ventricle, and then you're gonna have them in the fourth ventricle as well. Okay, and so in all these structures, you're gonna have the cord plexus, and that they are going to be creating the CSF from the blood. Okay, so once you fill up the lateral ventricle, the CSF is going to flow into the third ventricle. But in order for you to do that, you must be able to go through the interventricular foramen. Okay, a hole, right? And from the third ventricle, it will go into the fourth ventricle. In order for you to do that, you will have to go through the cerebral aqueduct. Okay, and then from the fourth ventricle, it is going to go into the central canal. Okay, central canal, which is in the middle of the spinal cord, central canal. And then from the central canal, it will go all the way back up to your brain. Okay, so this part is a little bit hard to visualize. Let me try to draw it out for you. So here is your brain, looking at it from the back. Okay, here's the brain, and then that would be the ventricles that we were looking at. The lateral ventricles goes into the third in the middle, and then into the fourth, and then it's gonna go down through your spinal cord. Okay, so in the middle, there's gonna be the central canal, right? So the CSF is gonna go down through the middle, in the middle. Now, we haven't talked about this yet, but you just kind of have to believe me for now, I guess. At the bottom of the spinal cord, it actually frails out, okay, like a ponytail. And so the CSF will come out, and then it will surround the spinal cord, okay? It will surround the spinal cord, and the CSF will flow back up to the brain, okay, where it will surround the brain. It will surround the brain. Remember, the CSF forms a cushion layer around the brain and the spinal cord, right? And so all that excess is going to drain right here. So you might remember that's where the dural venous sinus is. Okay, so from the central canal, you're going to go back to the dural venous sinus. Okay, and they will drain it back to the blood. All right, so this is basically a simplified version of the circulation of CSF. Okay, so just to be clear, these are the LV. This is the third and the fourth. Okay. So what happens if there is a blockage in this path? Okay? If the flow of CSF is interrupted, well, then you are going to have a buildup of CSF, and that could lead to a condition called hydrocephalus. Okay, hydrocephalus. Cephalus is the brain. Hydro is water. So water is building up in the brain. Okay? And in infant, the ventricles can enlarge due to the cerebral spinal fluid accumulation. So in this picture, in this scan, you can see the left two pictures are normal. Okay, and then with hydrocephalus, right, this is hydrocephalus. The buildup, the accumulation of CSF actually creates so much pressure that it's pushing on the lateral ventricles, and it's going to be damaging the brain tissues surrounding the lateral ventricles. Okay, and in kids, sometimes you might heard of babies that are born with really big heads, and that could be caused by hydrocephalus, because the baby's skull is still not fully converted into bones yet. They are still cartilage on some level, and so all that extra pressure is going to help push against the skull, and when it's still cartilage, it's going to expand, and of course that is not a good thing. So in hydrocephalus, you would have to try to drain the excess CSF by creating an alternate flow path called a shunt, and sometimes it carries to the abdomen, and there is going to be a pouch that the person have to carry to drain the CSF. But a buildup of CSF could happen if the CSF flow is interrupted. Maybe it's caused by a tumor, maybe it's caused by inflammation, and so on and so forth. So that's hydrocephalus. Next, we have meningitis. As mentioned earlier, meningitis is the inflammation of the meninges. It could be caused by bacteria, could be caused by virus. It turns out high school and college age students are at high risk of getting meningitis. You can get vaccinated against it, but there are different strains of meningitis, so you will have to get different shots to be protected from all of them, and OHIP only covers a few of those shots. If you want to get the maximum protection, then you might have to pay out of your own pocket. And the symptoms of meningitis include headaches, fevers, and a stiff neck. And if not treated quickly, a severe form of meningitis can lead to paralysis, coma, or even death. So to diagnose meningitis, the doctor can order an X-ray, CT scan, or a spinal tap. So a spinal tap involves a person lying on the examination table like that. They're going to spray some freezing agent in this area to kind of numb the feelings, and then they're going to inject a needle between your L3 and L4 and take a sample of the CSF. So your spinal your backbone rather. Your backbone actually has many different sections to it. So in the lower back region, they are called lumbar bones. So there are 5 of them. We named them L1 to L5. So between L3 and L4, they're going to inject a needle, and they're going to take a sample of the CSF, and they will culture it to see what kind of bacteria is causing the meningitis, and prescribe the treatment accordingly. Now, this looks like a scary procedure, and it definitely is, but it will not damage your spinal cord, because at this location, at the lumbar section, your spinal cord actually frails out like a ponytail, as we mentioned earlier, right? So if you try to insert a needle into here, then these things are just gonna move out of the way, right? It's like trying to poke through your hair with a needle. It's just not possible, right? They're just gonna move out of the way, right? So for that reason, the lumbar puncture, that's what the spinal tap, the same thing. This procedure will not likely damage your spinal cord. If you are a mother during labor, you might have received epidural. Epidural is like a local numbing agent, if you would, and that is also administered at the same location. They inject a needle into it, and they would pump the medication into it, and it will basically numb the pain during delivery. So that's it for the first part of the video. We will continue in the next one.